So the same thing, if the population grows, so this is the discussion part of the test. So if the population grows in size, as I said a few minutes ago, there will be a lot of mutations at the lower frequency. So you tend to observe excess of mutations at the lower frequency. And that's in the like a very left end of the frequency spectrum. So then in this case, both D-test and E-test are very sensitive type because this type of derivatives. Then you also can use H-test, but H will be ineffective because H is compare the intermediate, the average, because it's the average heterozygosity compare with the overall theta. So then another is like a population shrinkage. So it's population expansion and population shrinkage are like the events before the bottleneck and after the bottleneck. And so there's a lot of discussion or population size affect the polymorphism level. Actually, you can think of very simple example. You have a number of like 100,000 individuals shrink into like 10 individuals, and what will happen to the polymorphism level in the population. And this is the table from the review paper. I didn't talk about every single one, but I do talk most of them. I think the one I didn't talk is the FST and also the HKA test, but I think you can look up in detail if you need to understand. So basically today's lecture will try to, we can separate into two parts. One is focused on the coding sequence and using the synonymous, non-synonymous comparison to identify the positive selection, most positive selection for a certain genes from the genome or a particular size or like different size within a gene, which might subject to positive selection. And the second part is to utilize the population sample, the polymorphism data, and use the polymorphism data try to detect the signature of positive selection. But we are not only detect the positive selection, we also can know a little bit about the demographic, like a population like a size change in the population history. So that's the one we try to do, and those are the common methods we used in detecting like a signature of natural selection.